Welcome back to Dr. Liu's VCE Biology. Today's topic is regulation of biochemical pathways, as presented in the 2025 NHT Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. 3 separate solutions contained the same substrate and either enzyme A, enzyme B, or no enzyme. All other variables were controlled. The graph below shows the amount of product produced in each solution over 20 minutes. Both enzyme A and enzyme B significantly accelerate the reaction rate compared to the no enzyme solution, confirming they act as catalysts. Between the 15 and 20 minute mark, the rate of the reaction catalyzed by enzyme B remains constant, or shows a stable rate, indicating that enzyme B has not been denatured during this time. Since product formation occurs even in the absence of an enzyme, the reaction naturally proceeds at a slow rate. The rate of reaction is not zero. The data showing the amount of product formed over time does not provide any information about the three-dimensional structure of the enzyme's active sites. A competitive enzyme inhibitor that mimics the shape of acetyl-CoA would bind to the active site of the enzyme citrate synthase in the Krebs cycle. This competitive binding directly prevents acetyl-CoA from interacting with the active site. Because the inhibitor is competing for the same binding location, the active site, not an allosteric site, its presence effectively decreases the overall rate of the Krebs cycle. This presentation segment is dedicated to exploring the conception of enzymes as catalysts and the mechanism of enzyme inhibition. This section contains 2 multiple-choice questions, contributing 2 marks, 1.7%, to the total assessment. Question 6, graph interpretation, requires students to extract and compare data from a provided graph illustrating 3 reaction conditions: enzyme A, enzyme B, and no enzyme. The expected outcome is for students to formulate the conclusion that both enzyme A and enzyme B accelerate the reaction, act as catalysts, while recognizing that the reaction still occurs, albeit at a significantly slower rate, in the absence of an enzyme. Question 9, Enzyme Mechanism assesses understanding of competitive inhibition. Students must identify that the inhibitor functions by mimicking the substrate's shape, allowing it to bind competitively to the enzyme's active site. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.